So now we're going to be moving on with what seems like a pretty simple question, but it's going to lay the groundwork for a lot more complicated questions. The question is, how do cells sense insulin? Cells cannot directly sense glucose. They need another proxy. Sensing glucose would be too hard, and it's a fundamental purpose of each cell. So you don't want to have a specific glucose sensor. It's actually easier to make a protein that goes with another protein, and that protein tells you whether you want the cells to take up glucose or not. So in a sense, you have some sort of master area of the body that is determined with whether the whole body needs to take up glucose as a body or not take up glucose as a body. And that decides whether there's going to be insulin or not. Therefore, you do not need a glucose receptor on every cell. But the cells run on glucose. They need an insulin receptor on each cell. And the question of how that works is actually related to the question of how cells sense pretty much everything. How they sense all of these areas: sight, smell, taste, and growth, which is sensing time, and a mistake in this is cancer, mistake in growth. All of these involves sensing something. You have different first messengers for sight, smell, and taste from outside the cell moving inside the cell. For cancer, it's a little bit weirder, but in a sense, the passage of time is going on. The cell's moving through the cell cycle. It does that at certain steps in a 24-hour, roughly, cycle of time. When that gets messed up, when the cell's watch gets messed up and it keeps on growing, then it's sensing time wrong in a sense. It doesn't fit quite as well on the metaphor level, but on the biochemical level, it actually fits really well because it follows many of the same patterns. In fact, insulin sensing has a lot to do with growth in cancer signaling, because when you have sugar around, when you have insulin around, you're going to want to grow. And so those really do overlap. There are a lot of similarities between the four areas, and a lot of similarity between insulin at the beginning and cancer at the end. But insulin receptor we're going to talk about receptor enzyme signaling, and that includes growth signaling. And then we're going to move on to G protein coupled receptors, which have to do with a bunch of different categories. We're actually going to start with adrenaline sensing. How do you sense this hormone that is sent throughout your body? Then we'll move on to how do you sense photons, rhodopsin, how do you sense smell, olfaction, and how do you sense tastes? And then finally, we'll return to how the same principles involving growth signaling carry out with the cell cycle. It is kind of a different category, but these things are related, which is why you can go to the Fred Hutch Cancer Research Center. Do you know what one of the 3 Nobel Prize winners who works or worked at the Hutch got their prize for? Figuring out smelling. Linda Buck, lab right next door to mine. And actually figured out some of the olfaction stuff that we're going to talk about today. Why is she at a cancer center? Because there's actually a relationship between part 6 and part 7. Okay. So, after you ate a big turkey dinner, maybe during the holidays, and the thing that would be the fly in the ointment for the understanding that turkey contains tryptophan and therefore you get sleepy, right? Well, if you have friends who ate tofurkey, they would get just as sleepy as you if they ate just as much as you. It's not about the meat or lack of meat. It's about the carbs. It's about the digestion. So you had consumed a lot of carbs. And we're saying that that's what took the energy in. A lot of those carbs get into your blood sugar. Your blood sugar goes up. At the same time, the signaling molecule for blood sugar, insulin, also goes up. Insulin is a hormone. It's really more like a protein than anything else. It's not quite big enough to be called a protein by some definitions, but I definitely call it a protein. It's a protein hormone telling your cells to take up the sugar and turn it into energy. Very important and also very small. It's one of the reasons why people study it. But let's not talk about insulin itself. We talked about that early on when we were talking about proteins and things like this. Now we're going to talk about what senses the insulin. And so you have this top view and side view of the insulin receptor. You see there's a lot of stuff going on, but you have this right here. You have the insulin in red that binds to the receptor. It's a dimeric receptor, and honestly, There's a lot of detail if you're really wanting to get into it. What you need to know, dimeric receptor, insulin binds on the outside, it brings two domains together, and then signaling starts. This is how signaling is communicated from outside the membrane to inside, that there's a conformational change and two receptors come together. So the blue domains are inside the cell, and now we have the question of getting the signal throughout the inside of the cell, but we've already gotten it inside. Now the one thing I want you to notice down here, see all the little green P's. The green P's mean that there's phosphorylation going on, and that means you have some kind of kinase activity. You have phosphates being put onto new places. And so these are initial parts of the cascade, so they get phosphate groups. And if you're a cell, where are you going to get phosphate groups from? Let's get ATP. This is an important process. It's important enough to the cell. It doesn't mind burning some money to be able to signal. And so it's going to actually take some ATPs, and it's going to take phosphates from them. And it turns out that phosphate bonds, for the same reason that phosphate bonds are good for ATP, they're medium strength. And so that means that they hold together well enough to definitely not just fall apart on you, but they're hard enough to break, and in fact, they're hard for water to break, that they will persist, and they will only break when you want to, when you have like a ATP phosphorylase site that binds ATP and attacks it at just the right angle. It takes a specific geometry to break ATP efficiently. And then you have that cool phosphoanhydride bond, which has all those chemical characteristics that make ATP special. It also makes signaling special. So actually neurons have insulin receptors. The insulin receptor will tell the neurons where to grow. So it's actually important to the brain that the insulin will tell the neuron which way to go. Zach?
Is the insulin kinase similar to ATPase? Because I remember ATPase being reversible if you reverse the ion gradient. I'm wondering if the kinase is similar.
Yes. And kinases and phosphatases would be the ones that go in the opposite direction. It's easy to make a kinase or a phosphatase for the same reason. Breaking a phosphate bond requires a particular geometry for the nucleophile to get in between the protective sort of negative oxygens to get at the phosphate, the partial positive of the phosphate. So that's all organic chemistry. And it comes down to the fact that phosphate, because it forms PO4, and because it forms linked PO4s, will actually be very good for this. It's good for linking it to another phosphate. It's good for linking it to like aspartates or to asparaginins and other phosphorylation targets on the protein. If you have a nucleophile, you have a potential phosphate binding residue. Okay. So you can see that they've stained for insulin receptors, and there's lots of insulin receptors on these parts of the neurons. These are the parts that will grow. So insulin just isn't for burning energy, it's also for growing in a certain direction. So let's look at that blue kinase domain, okay? If you look at that, this is what it looks like when it's inactive, when there's no insulin around. When the insulin is around and it twists open, you see that there's an active part, and there's a pretty big difference right here. You see this blue loop completely opens up. Now, the blue loop you have, this aspartate right here, that's in the middle, and you see that aspartate is still right here. This shows you how much the loop moves. The loop basically hinges open around that. And you have these three tyrosines that are right next to each other on the loop, and they end up moving a large amount. So it's just a little conformational change outside, a dimer forming, two monomers coming together. And that twists and turns the proteins enough that this whole loop flips open. But then all of a sudden you have these three tyrosines exposed, and all three of them can be phosphorylated. So you have like a buy one, get two free kind of phosphorylation event. You have three places to put a phosphate. And this makes a pretty big chemical change. You've got a negative charge now, where before you didn't have, you had buried partial charges before. Now you have exposed negative charges. And this makes a pretty big binding interface for another protein to look for. There's a protein that looks for this loop to be like this and goes onto it, binds it, and that starts the whole cascade, okay? And we quickly move from atom land, where we see the atoms, to cartoon land, because this is really complex. There's this has 7 layers of complexity here. The quiz is not going to give me the 7 layers verbatim, but I want you to see the general trends of what's going on. And also, there are some things that show up here that are spoilers, because they will show up in other parts of this lecture as well. So for insulin signaling, the protein that senses that opened up loop, the phosphorylated loop, is called IRS1, and IRS1 is itself phosphorylated by insulin receptor. It binds, okay, and then the insulin receptor has a kinase site that will allow IRS1 to be phosphorylated. So you got all of these green glowing phosphates going around. I always imagine phosphate as glowing green, but if you know me, I'm a little biased toward that color. But you can imagine it as being like a green light for the cell to go on. And so IRS1, the phosphate makes a binding site for this other protein called GRB2, and that actually is a multi-protein thing that's actually anchored to the inside of the membrane somewhere. It doesn't really matter too much to us, but cancer biologists who study this, it matters a lot to. Because this protein complex has at its middle this protein RAAS right here. Do you all remember RAAS from where we saw it previously? Where did we see it previously? Mm-hmm. Mm-hmm. Yeah. Yeah. Anytime you have stuff being built, stuff growing, probably have RAAS somewhere in there. It's like the character that always shows up when something's going on. It's like the Nick Fury of, I have to think that metaphor through. If it had one eye, that would be really great. But it's always in the middle of things. And so it's a hub for this. And the other thing that's weird about RAAS is it binds GTP. And in fact, it turns out that normally when you look at RAAS and a cell is turned off, you see it bound to GDP. But when the cell gets turned on, the GDP gets kicked out and it binds a GTP when it becomes activated. Something about the active conformation binds GTP, and then after a certain amount of time, the GTP will be cleaved to GDP and the whole thing will turn off. Remember, desensitization is an important part of this whole chapter. And anytime you see a G protein, that's sort of a timer for the cell to turn itself off by binding a GTP and then slowly hydrolyzing it. It's actually not that different from the fact that you have potassium channels that are slow next to sodium channels that are fast. You can use a slow part of a process to turn off a process and let it go for a certain amount of time. It's like motion-activated light. They turn off by themselves after a couple of minutes. It's that same kind of thing. After a couple of minutes, it'll hydrolyze the GTP, and the whole thing turns off. For those couple of minutes when it's bound to GTP and it hasn't had time to hydrolyze it yet, it's in the active conformation, and that does all sorts of other things. Now, here's where we get to alphabet soup. There's one letter to pay attention to here. The alphabet soup letter is K, okay? That's the letter of today. If we have the Sesame Street, that's what they called it, like the letter of the day. Did they have that on Sesame Street? Am I remembering things right? I haven't watched it in a while. The K stands for kinase. Whenever you see K at the end of a protein name, that means you have a protein that's going to go around. When it's activated, it's going to go around and put phosphates on things. And actually, I have to do one other thing. Do you guys remember the Portlandia skit about put a bird on it? Okay, kinases are the put a bird on it of the protein, but they're putting a phosphate on it. Okay, instead of a bird, it's putting a phosphate on it. If somebody wants to ask Fred Armisen if he would film that for me, I'd write him a script. I don't think he'd say yes, though. Okay. But the thing is, once you get down here, you end up with a bunch of phosphates. And here you have one phosphate that gets phosphorylated. And it goes around and phosphorylates this other one. And then that one is able to actually move into the nucleus and turn on DNA that will program growth and all that. So you have these things. Phosphorylation means that you're sending a signal. GTP binding means you're sending a signal. GTP cleavage turns the signal off. And down at the bottom, you have more and more phosphate. Every level of kinase you have, each kinase can phosphorylate probably 10 different things as a minimum. So each time you reach a kinase level, you're multiplying your signal by 10. 2 levels of kinase, 100-fold activation. So notice RAAS. You've already seen it. RAAS is a G protein because it binds GTP, that's what it means. And it activates the cell in multiple ways. Certain cells, so the signal to divide goes through RAAS. The signal to take in insulin sends this signal in some cells. In other cells, a different first messenger will get you to RAAS. So RAAS is very important in studies of cell growth in cancer, as you've already seen if you look back at that Wells paper. The big difference between those were RAAS, I believe. And I believe that weren't they testing RAAS inhibitors of some sort? It was something in that pathway that they were inhibiting to stop the cell from growing, to stop it from being a cancer. What was it? Okay. Okay. Yeah. And that might be the surface protein that they were looking at. And so, yeah, we're looking at all parts of the pathway here. But the RAAS is deep inside the cell, and it's a central sort of connecting point for all sorts of processes. Here's what it looks like. Of course, no surprise, we've got the crystal structure of it. Inhibiting it is a big deal because inhibiting it can cause it in whatever way. So like there's drugs that can prevent the addition of a fatty tail. There's, because they bind GTP analogs might be able to block it, but it binds GTP so well, it doesn't accept second best. And so if you put in a GTP analog, it'll do a little bit, but it won't bind very well. They don't make very good drugs. Zach?
What happens if you just make something that binds GTP better?
Then you mess up the rest of the cell, probably, because GTP is one of the, I'm not sure. I'd have to look at this. That's getting into cell biology at this point. But that would be, I'm sure people have looked into it, because the one thing that I learned about cancer, and this relates to the overall emperor of all maladies too, by the way. When I worked at the Fred Hutch, everyone was talking about RAAS. It's like it does too much for us to understand, not like it does too little. And scientists, when they have something like that, boy, they want to write grants about it. So we've been trying to study it, and many things do actually work. There's an experimental drug that will bind a cysteine found in a mutant form of RAAS. If you have a mutation in RAAS, you're probably going to have a cancer from it because it is so central. So insulin receptor works as a tyrosine kinase, and there are similar inside domains. They often activate, so other tyrosine kinases look like this on the inside. On the outside, they can look like anything. You basically have a receptor domain that will bind whatever first messenger you need it to bind. So notice how all the red parts are different, but all the blue parts are the same. That's because this tyrosine kinase on the inside, all of these different receptors look like it. It looks like it's been modularly taken up, and you just put a different top on it. And like this leucine-rich domain binds a certain growth factor. This is a different growth factor. There's a lot of growth factor receptors because there's a lot of signals to send to make cells grow. Those signals don't have to look anything like each other. You just make a new red domain, put it on the outside of the cell, keep the same blue domain, and you've got a new signaling pathway possibility. This is another more recent hormone receptor. A lot of these are growth hormones, and so human growth hormone will be involved with something like this. This is another one that looks like what we were talking about, and you see that you have some interesting conformational changes going on. This is called JAK2, but when JAK2 dimerizes, when you have the binding of the growth hormone, it does this big conformational change where it like scissors open, and then you have the active protein has a very different conformation there. Lots of stuff to find out here. We're not about the details, but we're about the dimerization, conformational change, activation, kinases. You know, that's kind of the take-home message. David Goodsell, my favorite, has painted a RAAS pathway. This is a different RAAS pathway. But honestly, if you had told me it was the insulin receptor, I would not have been able to prove you wrong, because this is a growth factor that causes dimerization. And then here's GREB2, and then there's RAAS, and then there's GTP bound to RAAS, and it all looks pretty much the same. You still have RAAS at the middle of it all, and the proteins look the same. By the way, the RAAS is membrane bound. I said that I didn't think that that was very important earlier. Well, one of the drugs that we talked about prevents the fatty acid tail from being put on it. So I guess it is important, you know, it's that kind of thing. And you can see the fatty acid tail right here in the little red. Goodsell deliberately made it red because it is the heart of the whole thing. And then you see purple MAP kinases that are sort of cascading throughout the cell. Different levels of these kinases, and you can have 2 or 3 levels, and that gives you a signal amplification that is huge. So you have something like that. Bless you. So I want to ask you, so that you aren't just taking this in, we've had these 5 complexes, these 5 characteristics of signaling pathways. So somebody tell me one of them that this complexity that you've seen, there's a reason why this isn't just bind something and immediately send the signal. There's several layers to this. There's like 10 different layers where there could be just 2. This complexity gives you advantages. So hypothesize about the advantages you might get. Here's 5 clues. It gives you advantages in at least some of these areas. So tell me, what advantage does signaling complexity give you? The easy one can come first. So the first person to raise their hand might get the easy one. Zach.
The more modular it gets, the harder it is for viruses to take advantage of the contribution. It's like from an immunology perspective.
Right. So the modularity, how's the modularity foil viruses exactly?
Because the virus is going to propagate early in the environment that's able to different proteins involved in multiple pathways, and the virus can interact with that to contract best on both pathways.
Okay. Yeah. Yeah. So the modularity, switching stuff around, might confuse the virus in a sense, and like give it less of a place to grab onto in the cell and things like that. And yeah, that is a definite possible advantage. I think. Yeah, you can go with. I think modularity is where that one best fits.
It's kind of like a virtual machine.
Okay. Yeah. Yeah. And actually, you can do a lot of. Yeah. So sort of keeping the stuff away from. Yeah. You mess up one thing, you don't mess up everything. That's a good example. And actually, those metaphors, even the computer virus metaphor, works really well with regular viruses. The way the information is arranged. Mirka.
Also, you need to match for each molecule because once you send the new molecule and amplify the signal, so you get a cascade.
Okay.
So you have much of it, kind of it, but it's very specific,
Mm-hmm.
and cause amplification.
Right. Each layer of complexity adds amplification.
Right.
So you can definitely pull that one in. Yeah. Any other ideas? I've got 4 on the next slide. And there's one of them that is really, I never would have predicted. I'd like to see if you can hit on that. Oh, yeah. Yeah. So different first messengers require complexity for specificity. You need at least a different growth factor receptor for each different growth factor. And when you have a complex body that requires complexity at that level, you have complexity at that level. So yeah, that totally goes.
Another element is kind of talking about homeostasis and how it relates to desensitization.
Mm-hmm.
And once you establish a new baseline for the organism, you need the level of stimulant to reach it further.
Okay.
So if you're giving someone medication, try to go with, let's say, +2. Eventually that goes down to 0, you need plus, and now you get +2.
Yeah. And in a sense, it could be as fundamental as if you have desensitization, you need at least 3 conformations, which is complex. You can't just have on and off. You have to have off, on, and then on for too long, so I turn myself off, but I'm still waiting to be receptive to another on signal. Okay? There's probably a more efficient way to say that, but you can only do so much when you're standing up here. Anybody want, homeostasis is actually related to the one that I would never have gotten. It's kind of a weird way that homeostasis is related to it, though. So anyone wanted to give one more try? Zach.
The only other thing is because amplification, because a lot of
Mhm.
drugs that we give people don't actually directly affect anything. They affect like a hormone pathway that upregulates the homeostasis of the body.
Right.
So
Yeah, so more, and that could be related to integration as well, because you can have multiple effects if you have multiple. The integration works both ways. You can bring two signals together, or you can send two signals apart, and I think that relates to that. And so I think I do have this one, the amplification. You've seen a lot of kinases, and so the more levels of kinases you have, the more amplification you have. There's possibility of adding two signals together or downregulating other signals. I think this has to do with what you were saying. You can have multiple causes, and that actually relates to Hannah's as well. You can have multiple effects, more than one effect from just one receptor ligand information. You can have divergent effects where you have multiple signals. And in fact, insulin is an example of having multiple effects. I'll show you that one on the next slide. Here's a really weird one. It costs a lot of complexity to establish homeostasis. Reducing the noise in a cell, like being warm-blooded and keeping your temperature at plus or minus 1 degree Celsius, requires a lot more complexity than keeping it at plus or minus 10 degrees Celsius. There's a computational study that supports this. It says that if you have a 10-fold increase in temperature homeostasis, just like what I mentioned, going from plus or minus 10 to plus or minus 1 degree requires plus 10,000-fold molecules. Not all different molecules, but 10,000-fold molecules to play the different roles of keeping that tolerance down, keeping the error in temperature down. That's kind of astonishing. Maybe it's not 10,000, but that's a really huge number. And so it might be homeostasis requires complexity. Keeping things the same requires things to be complex, which seems kind of backwards, but that's what it says. Mirko.
I don't think it is the right way or the wrong way, but
Mhm.
it depends this organism. If you've got multiple processes, you produce less energy, so you're putting less work. But if you were to do just one big process,
Mhm.
lower the temperature, rather you need to put in more energy, so use more work, use more ADP. But if you break it down to more complexes,
Mhm.
then you actually do less work. So it's less.
Okay, so it's like reversible versus irreversible.
For the body.
Yeah, I'd have to think about that to completely put it together, but it seems at first blush that that could make sense. Biology wants to integrate. That's why calculus comes in useful sometimes. Integrating gets you more area under the curve, and that might be related to this. In fact, it probably is in the paper somewhere. But that requires calculus, and I promised that I would not have calculus in this class. Calculus is in that other class, so. This is an example of the divergent effect you can have from insulin binding. Here's IRS1. We start in the same place. But instead of going to RAAS, it goes to this other kinase, and it goes to this protein kinase B. Protein kinase B then phosphorylates this other kinase, which weirdly turns it off. But there's no reason, there's no rule that phosphorylating something is a structural change. And so phosphorylating can turn something off as well as on. And it just turns out it turns this kinase off. This kinase is always keeping this other protein off by keeping it phosphorylated. Like every time the phosphate falls off, it will put another phosphate on. But when it gets turned off, then the phosphate will get cleaved at a certain rate, and then this will become active. This is glycogen synthase. So if you remember your metabolism, glycogen is something that you want to increase glycogen synthesis when blood glucose is higher, because you want to take some of that glucose and save it for a rainy day. Because you have so many purposes for blood glucose, you have many effects of insulin, which can be different. For example, glycogen synthesis. And in fact, you can have even other ones. When glycogen synthesis is accelerated, protein kinase B can bind more than one protein. It does some other things, and we have a famous dotted line, so we don't know from this figure exactly how it does it. But it interacts with some other proteins that end up taking vesicles that include glucose receptors and putting those glucose receptors on the surface of the cell. All right? So you've got this thing happening. You got glycogen being synthesized. That turns off. and then glycogen is made. And then you've got one more effect. So this is a third effect that you have from it, is that this is effect number 2, this is effect number 3. You can have glucose receptors moving to the membrane. So this is very complex, but real life is even more complex. The main thing is 1 molecule, 3 results from this. That's what I said, at least 3 results. And if you think about it, protein kinase B can go around and it can bind other proteins. Maybe it binds other things. That could be a 4th, 5th, 6th effect. It's pretty easy to do that. And that's where this complexity comes from with this whole area. All right, and that ends part 4.